Okay, so good morning, Dr. Huang. Today we are going to present to you about wine fermentation and the microbes contained inside them and all of the information about wine fermentation. And my group has me, Dixon, and Wenyi. Alright, so these slides will be talking about what is wine fermentation and why did we choose this topic. So wine fermentation is the process by which grape juice or must is mean that crushed grapes with their skin, seeds and stems inside with it. So the grape juice or must is converted into wine. So during fermentation, yeast consumes the sugar in the grape juice and converts them into alcohol, carbon dioxide and heat. This metabolic process is known as alcoholic fermentation. The yeast strains used in winemaking are typically Saccharomyces cerevisiae, which are specially cultivated for their ability to tolerate the alcohol levels in wine and produce desirable flavors. The fermentation process can take anywhere from a few days to several weeks, depending on factors such as temperature and desired wine style. And why did we choose this topic is because we find this topic interesting and probably not much people are doing this genre of food fermentation. So out of our curiosity, we decided to make up our mind and chose this topic. Alright, now we are going to talk about what are the microbes contained in the wine, the taxonomy, the structure, characteristics, and its growth conditions. And we will talk about this on the next slide. Alright, so the first microbes that is present in the winemaking is called the Saccharomyces cerevisiae. So Saccharomyces cerevisiae, commonly as a baker yeast or brewer yeast, is a species of yeast that plays a vital role in various aspects of human life and industry. So here's a brief overview of Saccharomyces cerevisiae, its characteristics, uses, and significance. So Saccharomyces cerevisiae is a unicellular fungus that belongs to the kingdom fungi, as shown in the slides. It is a eukaryotic organism, which is shown that the domain is eukarya, with a relatively small genome, consisting of about 12 million base pairs. This yeast species is round in shape and reproduces asexually by budding, where a small daughter cell grows out from the mother cell. One of the most well-known uses of Saccharomyces cerevisiae is in baking. When used as a leavening agent, it ferments sugar present in the dough and produces carbon dioxide gas, causing the dough to rise. This process is essential in creating the airy texture of bread, pastries, and other baked goods. In addition to baking, Saccharomyces cerevisiae is widely utilized in the brewing industry. During beer production, yeast cells consume sugars from malted grains and convert them into alcohol and carbon dioxide. Through the process of fermentation, the specific strains of Saccharomyces cerevisiae used in brewing contribute to the flavor, aroma, and overall character of the beer. So that's why you see the characteristics. Saccharomyces is used in winemaking, cider production, whiskey, and it is unicellular, which is single cell. Beyond culinary applications, Saccharomyces cerevisiae have also been extensively studied in the fields of genetics and molecular biology. Due to its well-characterized genome and ease of manipulation, it has become a model organism for studying basic biological processes. Researchers have made significant advancement in understanding fundamental cellular functions by studying the genetics, metabolism, and signal transduction pathways of Saccharomyces cerevisiae. Furthermore, Saccharomyces cerevisiae has played a crucial role in the development of recombinant DNA technology and the production of various pharmaceuticals through biotechnology. By introducing foreign genes into the yeast cells, scientists can harness their cellular machinery to produce valuable proteins such as insulin, human growth hormone, and vaccines. This application has revolutionized the field of biopharmaceutical production. And the study of Saccharomyces cerevisiae has also contributed to our understanding of human disease. Many genes and cellular processes in yeast are conserved across species, including humans. Thank you. Alright, so in this slides, we're going to talk about Oenococcus oini. It's another microbes present in wine fermentation. So Oenococcus oini is a bacterium commonly found in wine production. It plays a crucial role in the process of malolactic fermentation, which occurs after the primary fermentation carried out by yeast. Malolactic fermentation is the conversion of malic acid into lactic acid. It provides a smoother and more complex flavor profile in wine. Oenococcus oini belongs to the lactic acid bacteria, which is known as LAB group, and is a gram-positive, non-motile, and non-sporulating bacterium. Its name oini is derived from the Latin word oinum, meaning wine. This bacteria is naturally present on grape skins and can also be found in winery environments, including barrels, tanks, and equipment. During malolactic fermentation, Oenococcus oini consumes malic acid, which is naturally present in gas, and converts it into lactic acid. This process lowers the wine acidity and contributes to its overall flavor and stability. Additionally, the production of lactic acid by Oenococcus oini helps inhibit the growth of harmful spoilage microorganisms, improving the wine's shelf life. This microbes is highly adaptable to the challenging conditions of wine production, including high alcohol content, low pH, and the presence of sulfur dioxide, which is commonly used as preservative in winemaking. Its ability to tolerate these conditions is attributed to various factors, such as the production of enzymes that enable it to utilize specific nutrients present in the wine environment. Winemakers often rely on inoculating their wines with selected strains of Oenococcus oini to ensure a controlled and consistent malolactic fermentation. This practice allows winemakers to achieve desired flavor profiles and enhance the quality of their wines. The use of specific strains can also influence the production of secondary metabolites, such as volatile compounds, which contribute the aroma and sensory characteristics of wine. Furthermore, the application of modern biotechnology tools such as genetic engineering and genome editing techniques holds promise for the development of tailor-made strains of Oenococcus oenis. These advancements may enable winemakers to optimize the fermentation process, produce wines with unique flavors, and address challenges associated with changing climate conditions and market demands. And I need to mention about the growth conditions. To produce Oenococcus oenis, you need an optimal amount of 20 to 30 degrees Celsius, only that can grow. And the structure of it is ellipsoidal to spherical, and they grow in chains of bread. Alright, this is the third and last microbes that contains in wine fermentation. It is called the Botrytis cinerea. So what is Botrytis cinerea? It is a fungal pathogen that can have both beneficial and detrimental effects on wine fermentation. This versatile fungus is commonly known as noble rot and is responsible for the production of some of the world's finest dessert wines. However, it can also cause significant damage to grape crops and spoil wine if not properly controlled. In favorable conditions, Botrytis cinerea infect rice grapes, particularly those with thin skins. It forms a grayish mold on the grape berries, which punctures the skins and allows the fungus to penetrate the food. This process initiates the development of noble rot, where the fungus partially dehydrates the grapes by removing water content while leaving behind concentrated sugar and flavors. For winemakers that are seeking to produce sweet botrytis wines, this natural occurrence is highly desirable. The noble rot imparts unique characteristics to the wine, including flavors of honey, apricot, and dry fruits. The concentrated sugars provide sweetness and complexity, resulting in a lustrous and aromatic wine. Once the grapes are harvested, they undergo a meticulous sorting process to remove any undesirable rot or mold. This step is vital to ensure the quality of the final wine. After sorting, the grapes are gently pressed to extract the juice, which is then fermented. And Botrytis cinerea can survive under 10 to 20 degrees Celsius compared to the other microbes that I've mentioned. At its structure, it's large oval, spherical cornutia. It is either simple or branched. Thank you.
Hello guys, my name is Dixon, and I'm going to talk about the backstory of winemaking. In recent years, vessels has been discovered that contain the remains of wine with an age close to 7,000 years. It is unclear in ancient times humans accidentally stumbled across fermented beverage like wine or beer, or it is invented purposely. What is the fact is that since then, alcohol beverages have been a part of diet and culture of many of the civilizations that have come before us. The typical example of beer and wine are an example of many other drinks resulting from the action of yeast. Aside from these two types of beverage, companies have developed numerous fermented foods and non-alcoholic beverage by using traditional way or commercially manufactured. In addition, the traditional alcoholic beverage that I mentioned just now is produced from fruits, berry, and grains. Due to the climate condition, each region have condition and preference to the maintenance of these products. Fermentation is a well-known natural process used by humanity for thousands of years with the fundamental purpose of making alcoholic beverage as well as bread and other products. By using the point of view from biochemical, fermentation is a process of central metabolism in which an organism converts a carbohydrate such as starch or sugar into an alcohol or an acid. So, how was the fermentation discovered? Fermentation process was spontaneously carried out before the biochemical process was fully understood. In 1850s and 1860s, the French chemist and microbiologist Louis Pasteur became the first scientist to study fermentation when he demonstrated that this process was performed by living cells. Fermentation process to produce wine, beers, and ciders are traditionally carried out with Saccharomyces cerevisiae strains, the most common and commercially available yeast. They are well known for their fermentative behavior and characteristics which allow to obtain the products of uniform and standard quality. Many other important industrial products are the result of fermentation, such as yogurt, cheese, bread, and coffee. Yeast also play a key role in wastewater treatment or biofuel production. So, like what I just mentioned, primary yeast species used in winemaking is Saccharomyces cerevisiae. And yeasts are responsible for alcohol fermentation, where they convert the sugar present in grape juice into alcohol, carbon dioxide, and other byproducts. So let's see how they works. Take a look for the figure number 1 at the right hand side, which is the central metabolism of fermentation in yeast. So first step, which is the sugar metabolism. Yeast metabolize glucose and fructose, which is the main sugar in grape juice, through a process called glycolysis. This process breaks down sugar into pyruvate and produce energy in the form of ATP. After glycolysis, pyruvate is converted into acetaldehyde and then further reduced to ethanol, which is the alcohol found in wine. This conversion occurs through the action of enzymes such as pyruvate decarboxylase and alcohol dehydrogenase. Carbon dioxide production, which leads to formation of bubble in sparkling wine, and it helps to remove some unwanted compounds from the wine. Come to the first example of microorganism is applied in the winemaking process is Saccharomyces boulardii. This yeast provide a strong flavor intensity due to the presence of many acetic ester that generate in the process. It have an ability to settle the bottom of fermentation vessels, help the wine become clearer and sediment. In later steps, this yeast reduce the work when come to separate clear wine from yeast sediment. In another hand, Saccharomyces cerevisiae, also known as wine yeast or brewer yeast, play a crucial role in enhancing the fermentative process and wine quality. So, the active dry yeast is employed in wineries all over the world. This microorganism metabolize the sugar present in the grape juice into pyruvate molecules and generate energy for further usage. In some winemaking practice, this microorganism may also promote malolactic fermentation. So malolactic fermentation is a secondary fermentation process conducted by lactic acid bacteria, such as Enococcus ini, which is convert malic acid into lactic acid, which contribute to the overall flavor and stability of the wine. It is also a biocontrol agent that produce toxins throughout the winemaking process to prevent the growth of spoilage microorganism. Is there any impact of microorganism towards human? The answer is yes. The Saccharomyces cerevisiae, which is the brewer yeast that is commonly used in food fermentation, has been recognized as safe by the European Food Safety Authorities. It possesses various benefits and has been associated with positive effect on human health and well-being. Understanding the mechanism and application of this yeast is important. One well-known health benefits of this microorganism is its probiotic effects. The probiotic effect of the yeast can help to prevent and treat the intestinal disease. Furthermore, this yeast has the capacity to bind and detoxify mycotoxins through its cell walls, offering a protective function against these harmful substances. Overall, Saccharomyces cerevisiae has multiple beneficial functions, including probiotic effects and mycotoxin detoxification, which is impact on immune system. This yeast has been shown to have immunity regulation effect, meaning it can help to regulate and enhance the functioning of the immune system. Hence, exploring these positive effects and understanding the mechanism behind them can further expand the utilization of yeast in various applications and promoting human health and well-being.
Okay, so let's talk about the future of the microbe. First is the human microbiome. There's ongoing research on the human microbiome suggests that our understanding of the microbiome community within the body is progressing. This could lead to the development of personalized medicine and a unique microbiome profile for disease diagnosis and treatment. In recent years, a research on treatment for atherosclerotic cardiovascular disease has focused on lowering LDL level, which is the lower density lipoprotein cholesterol, and addressing the transitional risk factor, and resulting in a significant reduction in vascular mortality. This research showed that the gut microbiome acts as an endocrine organ, producing a bioactive metabolite that can impact the atherosclerosis. And this review examined two gut microbiome metabolite or the pathway impacting the atherosclerotic cardiovascular disease. And they also discussed the effect of TMAO, bile axis and aromatic amino acids metabolite on the atherosclerosis progression. Gut microbiome enzyme and receptor could be targeted for the new drug. Although it needs more research on this mechanism to eliminate the drawback, but it still have a good prospect on this research. Next will be the microbial bioremediation. It is a field that utilizes microorganisms to degrade or detoxify pollutants. Ongoing research in this area is leading to the development of more advanced technology. Scientists are gaining a better understanding of microbial pathway and are working on engineered microbial strains that are specifically target and degrade pollutants. This advancement has a potential to provide innovative solutions for environmental cleanup, allowing for more efficient removal of contaminants from soil, water, and air. One approach involves stimulating the growth of microbes through the addition of nutrients to contaminated soils or using biofermentation technique in specific cases where microorganisms are needed to address contaminants. Intrinsic bioremediation, on the other hand, is effective in soil and water environments, particularly in underground petroleum tanks where microorganisms can remove toxins and clean the tanks. Overall, microbial bioremediation holds prospects for addressing environmental pollution and restoring contaminated environment. Although in the next 10 to 15 years, our future of microbe has good prospect, however, there still have challenges the scientists might face. As in the world of microbial technology, ensuring safety regulation and ethical consideration is crucial. As advancement continues, it is important to assess the risks associated with genetically modified microbes, engineered strains, and microbial therapies, and establish or update the regulatory framework to ensure their safety implementation. Understanding microbial interaction and overcoming technical limitations are still remain ongoing challenges which require further research and development. Filling knowledge gap in microbial function and their interaction is also essential for optimizing microbial application. Public perception and education play a vital role in fostering informed discussion and decision making, addressing concerns, and building public trust. By addressing this challenge, we can responsibly adopt the full potential of microbial across various fields. Okay, so let's recap on what we have presented from previous slide. So first we have talked about what is the wine fermentation and how it becomes the alcohol. Then we have shown a few examples of the microbials contained in the wine, such as the Saccharomyces cerevisiae, which act as the primary yeast in wine fermentation, and Onococcus onion, which make the wine more smooth when we drink, and also the Protidis cinerea, which provide a unique flavor and aroma to the wine. Then we have talked about the background story of the winemaking, on how it's been discovered, studied, and developed by scientists. And after that, we discuss on how the microbe work in the wine fermentation, like the role of the yeast during the fermentation and what is the chemical reaction included with detail. And moreover, we have explained the impact of changes on human due to the microbe, which is mostly about how we use microbe to help or benefit the society. And last, we have talked about the future prospect of the microbe in the next 10 to 15 years, and which can further develop the use of the microbe and the challenges ahead we need to face when developing. In conclusion, microbe play a vital role in the production of wine, as it contributes to its flavor, aroma, and overall quality. Throughout the winemaking process, these microbes interact with grape juice, while initiating fermentation by convert sugar into alcohol, while other yeast and bacteria contribute unique flavor and complexity. Managing microbial population is crucial for consistency and quality, which can be achieved through techniques such as temperature control. Since microbes are essential contributor to wine complexity and quality, so winemakers need to carefully balancing desired and undesired microbial activity for consistent sensory profile and long-term stability, which can prevent spoilage, which can be achieved through the technique like sterile filtration, cold stabilization, and so on. Ongoing advancement in research and technology can further enhance understanding and exploration of a microbial contribution in winemaking. For example, like exploring the impact of indigenous yeast, although their unpredictability poses challenge.